project presentations always involve an interdisciplinary collaboration between two PIs or a PI and a postdoc. And in our meeting, it's going to be a cell biologist and a computational modeler or data scientist. And the way the presentation works is usually the biologist describes the biological aspects of the project for 20 minutes. And then the modeler talks about the modeling aspects for 20 minutes. And then we have two of our invited experts who should come up on stage when Stephanie comes, and they will be interacting with the presenters after the 40-minute presentation. We have 20 whole minutes left for that interaction and for questions from the audience. So we'll let the project presenters interact with the experts for 10 minutes or so, and then open it up for questions in person and virtually. What we insist upon is that this work is in process. It's not published work. And we tell the project presenters, imagine you're going down the hall and just talking to your colleagues and letting them know what you're doing and getting their feedback. So this is very much in process work, not done work. So that's all I want to say. I'm going to invite Stephanie to come up and start it off. And the two experts, we've got live.
All right.
Okay. So for the people that are watching virtually, we can control that camera. We start with it zoomed in on the podium, but when we go back to the modelers on stage, we're going to zoom it back out and then again to show the audience for questions. This requires me to come up and tap a few buttons. So just pretend that I'm, not there, just ignore me. Okay, so right now, is that what it's on? Yes. I'm going to make it. So this should zoom into, yeah.
Oh, yeah. Oh, no, I can see myself now. Okay, great. All right, so let's get started. Thank you to the organizers for giving us this chance for Tom and I to talk about our collaborative project. I'm Stephanie Woo. I'm an assistant professor at the University of California Merced. I'm the biologist half of this project presentation. And today I will be talking about the development of this tissue that we're seeing in green here, which is called the endoderm. I am a cell and developmental biologist. So I'm interested in how cells get together to form tissues. And in particular, I'm interested in how dynamic behaviors like cell migration and cell adhesion and cell-cell interactions are developmentally regulated during tissue formation. And the particular tissue that my lab focuses on is, so this is weird. The particular tissue that my lab focuses on is called the endoderm. This is an embryonic tissue that arises very early in embryonic development. It's the tissue that will eventually give rise to the epithelial lining of all of these organs listed here. So the organs of your digestive tract, your respiratory tract, and some other organs like thyroid. So this is the endodermal contribution in the adults. But if we rewind back in time to an early embryo, so this is a diagram of just some generic embryo, you'll see that the endoderm, so that endodermal tissue lies on the inside of this embryo. So the endoderm forms the innermost layer of the early embryo. But the cells that make up the endoderm do not they do not arise from that inner position. They actually, in most embryos, are specified on the surface of the embryo, and then those cells have to somehow make their way to this inner layer. And so lots of different organisms have developed different strategies to place those endodermal cells on the inside. So this occurs during a really important time in development called gastrulation. And so the model organism that my lab uses is zebrafish. And zebrafish use active cell migration to internalize their endodermal cells and again, place those cells on the inside of the embryo. So this is really interesting to us because this means that there are at least two major transitions in the way that cells, the endodermal cells behave. So first, you have this sort of epithelial to mesenchymal transition that allows these cells to begin migrating and make their way to the inside of the embryo. And then, like I said before, eventually the derivatives of the endoderm are the epithelial layers of these organs. And so these actively migrating cells that now make their way to the inside, they have to undergo a second transition, a mesenchymal to epithelial transition, to differentiate into the epithelial layers of these organs. And so this makes the zebrafish endoderm a really nice model to understand the decision-making process between these transitions in cell motility and cell adhesion. Okay, so that's just a general overview of endoderm development. Let's drill down to look at what this looks like specifically in zebrafish. Like I said, the endoderm is an embryonic tissue, so it's specified really early in embryonic development. In zebrafish, this happens at about 5 hours post-fertilization. So for those of you who have never seen a zebrafish embryo, at this stage, the embryo just looks like a ski cap sitting on top of a giant ball of yolk. So the yolk here is depicted in yellow. And the endodermal cells are specified at what you can think of as the edge of that imaginary ski cap. So at the margin between the embryo proper and the yolk. So soon after these cells are specified and know that they're supposed to be endoderm, they undergo that ingression process where they become migratory and they internalize. So they flip under their neighbors to form an internal layer. However, in zebrafish, this inner layer is, at least initially, not coherent. So as soon as these cells internalize, they actually disperse away from each other. And so forming this discontinuous salt and pepper pattern of endodermal cells across that inner surface of the embryo. However, this dispersal is transient, so eventually these cells will converge back together on one side of the embryo, and initially they will just form a flat endodermal sheet, and then that sheet will eventually differentiate into the different organs that the endoderm contributes to. So one of the advantages of zebrafish as a model system is that it's very amenable to live cell microscopy. So this is a movie that I'll start in a second so this is one of the transgenic fluorescent lines that we use a lot in the lab. So it's using the endodermal SOX17 promoter to drive expression of GF. And so what I want you to focus on are sort of these dimmer cells out here, and you can see that they're already dispersed. These are the endodermal cells. This bright cluster here are a population of non-endodermal cells that just also happen to express SOX17. They're also super interesting to us, but they're not the focus of today's talk, so just try to ignore them for now. So as I play this movie, you'll see that the endodermal cells are very dynamic and migratory. And towards the end of the movie, we'll see that convergence process as they converge on the middle of our field. So we can break down these different migration behaviors into two broad phases. So again, there's an initial dispersal phase that spreads out these cells across the inner surface of the embryo. And from work in my lab and other labs, we know that this dispersal is characterized by directionally random migration. So as these cells are migrating, they have very low directional persistence. And as you can see, they have very little to no adhesion or strong interactions with each other. So we can see that again in these higher magnification movies. of cells bouncing around. And so you get this feeling that they're really just sort of free agents and undergoing single cell migration. And then, so later on, they do converge back together, and that's characterized by, so instead of random migration, now they have highly directional migration. So they move with increasing directional persistence towards one side of the embryo. Well, they will adhere to each other to form this endodermal sheet. And again, we can also visualize or image this process at higher resolution. So for the purposes of today's presentation, I'm going to mostly focus on this dispersal stage. So when I was a postdoc, I and some other groups showed that, again, this dispersal behavior is driven, at least in part, by this intrinsically low directional persistence that these cells have. So they tend to switch their direction of migration quite frequently. And when I was a postdoc, I showed that this intrinsic lack of directional persistence is driven by signaling from the TGF beta molecule nodal, activating the Rho2. What I'm going to tell you today is that we think there are also extrinsic factors driving the dispersal of these cells, specifically cell-cell interactions and specifically inhibitory cell-cell interactions. So we think that as these endodermal cells are dispersing away from each other, they are undergoing a type of behavior called contact inhibition of locomotion, or CIL for short. So CIL has been observed in many different cell types under many different contexts. And there are, so these inhibitory interactions can either occur between cells of the same type. So you can have homotypic CIL, or they can occur between cells of two different types, or heterotypic CIL. We can further break down contact inhibition locomotion into two broad subcategories based on the actual, based on the specific response of a cell in response to colliding or contacting another cell. So in type 1 CIL, a cell will respond to colliding with another cell by changing its direction and moving away. And so we call this active repulsion type 1 CIL. In type 2 CIL, what happens is that when a cell bumps into another cell, it sort of just stops or pauses. It's characterized by a brief cessation of movement. And then just through random deflection, that pause will be resolved and the cell will start migrating. So the first thing we wanted to ask was, do endodermal cells truly undergo CIL, and can we characterize what type? So here is a movie of endodermal cells migrating. If you watch any of these cells over time, you'll notice that as cells bump into each other, they tend to change direction and migrate, and sort of push off and repel each other. And so we can look at that a little more clearly in these still images. So these are still images taken from this exact same movie, but now it's been pseudocolored. So these cells are expressing a fluorescent actin reporter. So what we're looking at is the amount of actin content within the cell at these different points in time. And so what we noticed was this very characteristic response, or a sequence of events where initially upon colliding with another cell, you get this rapid loss of actin content at the site of contact. This is followed by membrane retraction of those contact sites and a reorientation of that actin-rich leading edge. And that seems to be what allows cells to push away and move off in different directions. So based on these observations, these observations suggest that endodermal cells are undergoing type 1 or active repulsion type CIL. So of course, as a cell and developmental biologist, what we're really interested in, what we really want to do is drill down into the molecular mechanism that cells are using to both recognize each other and initiate this repulsion event. And so if you're really interested in the details of that mechanism, Jessalyn LaBelle is an incoming graduate student in my lab, and she has a poster presentation. She'll be at the poster session later tonight, so you should definitely hit up her poster if you want to learn more about that. But so briefly, what we think is happening in these cells is that they are using a cell surface receptor called FA2 to recognize. This is a contact dependent cell surface receptor. So we think this is what they're using to recognize that they've hit another cell. And we think that this signal is being transduced to the Rho GTPase Rho A, which then coordinates these responses of both membrane retraction and leading edge reorientation. So what that means is that we can either by manipulating the activity of Rho A or the receptor FA2, we can sort of turn on and off CIL responses. So this is an example of cells expressing dominant negative Rho A. And in this case, I don't know if I can replay that movie. Here we go. So in this case, you'll see this cell here bump into that other cell, and it didn't care. It just kept going in the same direction. Similarly, if we inhibit the FA2 receptor, we see similar types of interactions. So there was a cell that just glided past another cell and didn't change its direction in response to collision. And this was using a small molecule inhibitor of the FA2 receptor. So either by turning off Rho A activity or the activity of the receptor, we can block this CIL response. And so that allowed us to ask what contact inhibition is good for. So how does this affect overall endodermal migration? So in either case, so again, we're using either using a dominant negative mutation to block Rho A activity or treating embryos with the FA2 inhibitor dasatinib. And in both cases, we see an effect on the patterning and positioning of cells. And so these are zebrafish embryos at about 10 hours post-fertilization. So at this point in time, cells are sort of maximally dispersed across the embryo. And not only are they widely distributed, but I hope you can appreciate that they also have this very highly ordered, very regularly spaced pattern in terms of their cell positions. If we turn off CIL, either with dominant negative Rho or with dasatinib treatment, we turn this very regular pattern into these irregular clusters interspersed with cell-free gaps. So we introduce, so without these inhibitory interactions, you get a lot more irregularity in the placement of these cells. We can see that. So for example, we can also see that if we look at the distribution of nearest neighbor distances. And so for example, turning off CIL by expressing the dominant negative Rho A mutant, we get a lot more variability in those nearest neighbors. Okay. So this was really interesting to me because it reminded me of a bunch of other contexts in which cells have this very regular arrayed spacing. So things like cells in the retina making retinal mosaics, the highly ordered tiling of dendrites of sensory neurons in the skin, and the placement, the highly ordered placement of hemocytes or blood cells in the Drosophila embryo. And all of these, each of these examples I'm showing you, this highly ordered spacing is driven by mutual repulsion between these cells. So we think that endodermal cells are using the same mechanism of using self-avoidance to generate this highly ordered pattern. And in other contexts, especially in these sensory systems, this highly ordered pattern that's driven by self-avoidance is thought to be a mechanism to maximize coverage while minimizing overlap. So the difference though between these examples and the endoderm is that for these cells, this is sort of the final position or the final pattern that these cells are making. Whereas for the endoderm, this is a transient state, right? So after a couple hours after the cells position themselves here, they're going to converge back together into an epithelial sheet. So then what is the point then? Why poof yourself out if you're just going to smush back together? And so we're still working on an answer to that question, but we have some preliminary data that I think is pointing to an answer. So as I said before, we have good preliminary data suggesting that the receptor, the cell surface receptor FA2, is this receptor that is involved in initiating CIL. And so we're currently generating a CRISPR knockout of the FA2A gene in zebrafish. So we're creating a transcription start site deletion. And so this is very preliminary data, just showing what we call our crispins. So these are embryos that have been injected with CRISPR constructs, but they're not necessarily, it's not necessarily a stable mutant line, for example. But we did notice that some proportion of these injected embryos have discontinuities or holes in the endodermal tissue. So these are embryos at 24 hours post-fertilization when the endoderm should be organized with the pharyngeal endoderm in the head and the gut tube along the trunk. But it should be a continuous, coherent epithelial layer. And in some of these embryos with FA2 deletions, it appears to be discontinuous. Okay. So putting everything together, again, endodermal development can be characterized by both dispersal and convergence behaviors. We previously showed that this dispersal behaviors can be driven by intrinsic factors, so this intrinsic low directional persistence. But I think hopefully today I've convinced you that there are also extrinsic factors, namely FA2 and ROA dependent contact inhibition locomotion that is contributing to this dispersal. And these inhibitory interactions not only are promoting this dispersal, but they're also regulating this highly ordered spacing between cells. And we think that the spacing regularity is important for subsequent coherence of that epithelial tissue that's going to form through convergence. And so sort of that's where the modeling side comes in. And so with computational modeling, we hope to answer these two questions. So like I said, we think there are both these intrinsic and extrinsic factors that promote the dispersal of endodermal cells. So which is the best mechanism to achieve this, or do they both, are they both equally important? And also, again, so what is the significance of being evenly distributed to subsequent convergence and sheet formation? Okay, and so with that, I will hand it over to Tom. Okay.
Hello.
All right, yeah, I don't know what happened.
Yes,
after
it's
this.
okay. I got you. All right.
There you go, Tom. I'm going to spotlight you, and then if you're happy to share your slides.
Yeah.
I will mute us on this end. Does that work, Toby?
There we go.
Great. Is that all okay? Can everyone hear me? See the slides?
Absolutely. And we can see your first slide, which says questions.
Great. Okay, so I'll quickly start just by repeating those questions of ours that Stephanie has already introduced. So the questions that where we think modeling can be useful. So firstly, it's really what is the best mechanism or the best strategy for these endoderm cells to distribute and disperse evenly over the embryo. So from this starting position where they're all arranged at the edge of the enveloping cell layer like this, into a more evenly spread arrangement here. And we're mainly going to be looking at two competing strategies. The first, the contact inhibition of locomotion that Stephanie talked about, and also this non-persistent migration, so where cells are just moving around, changing their direction randomly. The second question again there is why cells are doing this. Why do the cells want to disperse like this when the next stage is just to reconverge? So we have cells which at this point, once they've dispersed, they realign. So we wonder then, as Stephanie mentioned, whether that's to do with getting the right distribution of cells to the dorsal midline, so getting the right cells to the right place at the right time. So to approach this with modeling, we've just made a simple agent-based model of the embryo. So we start with a sphere, which we say is the same size as the embryo. We put cells on the surface of the embryo, which again, we match to the dimensions of the cells from the data. And then first we make them move in straight lines along the surface of that sphere. And just as a slight digression, just so we're all thinking in 3 dimensions, really, I'll remind you, it's not always intuitive what it means traveling in a straight line when you're on the surface of a more complex geometry. So when on the surface of a sphere here. So here we have a map of the world, and we might be tempted to draw a straight line like this from Cambridge to Chicago, but in fact, the straight line between those two places, the path, which is the shortest total distance, is this one here. And that's, of course, because that 2D projection isn't very good at really showing us the 3D-ness of the problem. And it becomes a bit more obvious if you try and make it look at 3D. And if you rotate it like this, it makes it even more clear. So these straight lines, we know how to make. Mathematicians call them geodesics. So we make the cells in our simulated embryo travel along these straight lines. And next, oh yes, sorry, we take the velocity, the kind of average velocity that we see from the data as well. And then we want to make the cells travel in not straight lines, so more this random low persistence motion like this. And we can do that simply enough just by at each step in the simulation, we can add a small angle to the direction of movement. And we do that according to these equations. We add an angle which is randomly selected from a Gaussian distribution, where we've just set up the standard deviation of that Gaussian so that we can define this notion of a persistence length, which is really the length that you will travel on average before you've turned 90 degrees. So it's some idea of how far you travel in a straight line. The next thing to implement in the model is contact inhibition, of course. So this is a simple idea that when cells collide, they repolarize, and in the model, they move off in the directions which is directly opposite to the point of contact. And something which perhaps takes a little more thought is what to do when you don't have this effect. So if cells, they can't pass through each other at first, we set that they can't pass through each other, and then cells will be stuck here at that point of collision, unless we have this low persistence length. So if they're also changing direction randomly so that they'll collide and be stuck, but eventually they've repolarized in some way so that they can unblock themselves. Very rarely we do see in the data cells that manage to somehow crawl over or under each other. So we implement that in the model with some probability. Cells can just pass through each other like that. The last thing to tell you about in the model here is epibole. So in the bright blue at the top, we have the enveloping cell layer, and that's migrating down to cover the whole embryo. And that's limiting where the cells can travel for this part of the simulation. They can only travel where the enveloping cell layer has reached. So just the bright blue, like this. So a general simulation we set up with the same, roughly the same number of cells as we see in the embryo, starting in that starting configuration around the edge, and simulations progress like that. And the next thing we need, because we're interested in this dispersal and this homogeneous spacing of cells, we need some kind of measure of that heterogeneity. And we take a simple measure from this paper here, where you cut up the surface of the sphere into sections of equal area. And then if they were homogeneously distributed, we'd know how many cells we'd expect in each of those segments. And then we can just sum up all of the differences of what we actually find in those segments and normalize. And that gives us a very simple measure of heterogeneity, where if you're very evenly spaced, your measure is close to 0, like that. And if you have a very heterogeneous distribution of cells, for instance, here, it's kind of the worst one you can think of, where all of the cells are in one place, then you have a measure of heterogeneity, which is close to 1 there. So we can run simulations like so, where we can measure all along time how the heterogeneity is changing. And you can see that in this simulation, cells are, the measure is capturing how the cells are spreading over the embryo and creating this more homogeneous distribution. And now we can get back to that original question of comparing these different strategies. So these first results, we have the case where there's no contact inhibition, and we're comparing 2 persistence lengths. So one short persistence length and one long persistence length. So cells are traveling far in straight directions, not changing their orientation very often. You can see that the long persistence length does better at first, but then at some point it starts to do worse than the low persistence length case. So we can watch the simulations to see what's happening there. Here we have the low persistence length case, and you'll see by the time we get to this kind of 8-9 hour time point, the cells have been moving backwards and forwards and changing direction so much that they still haven't covered the distance of the embryo. They haven't managed to reach the poles there. We fast forward to 25 hours, you can see eventually cells are doing better. And so as you get to the later time point, it's continuing to improve and cells are finally reaching those empty spaces. Whereas the case with a long persistence length, cells start to do better at the beginning and reach further places around the embryo, but you get these aggregates forming, these build-ups, these aggregates where cells are stuck, and because they have this long persistence length, they're not resolving those aggregates, they're not changing their direction quickly enough. And if you zoom forward to later in the simulation, you can see these aggregates are really persisting and are what are causing this this worsening of homogeneity. But if we look at the case with contact inhibition, we get a very different result. That firstly, again, when cells have a long persistence length, they do much better at the start, but in this case, they continue to do better, and that's obvious in the simulations when you watch them. Again, at lower persistence length, the cells are slow to reach the poles. But in the case of a long persistence length, you have the advantage still that cells can travel long distances over efficiently over the surface of the embryo, but they're not forming those aggregates, of course, because the contact inhibition is stopping those aggregates forming. So this is just to show that the numbers we've picked are nothing special, that over more of parameter space, you get this uniform patterning, which I think clearly shows that if you want to quickly, efficiently disperse over the surface of the embryo, then a good strategy is to have contact inhibition and with long persistence length. So coming now to the second question, which to remind you was this question of what is the importance of this starting distribution? Why are you doing this when you're just going to reconverge here? As soon as you start to model this question, you realize that actually the first question that you have to answer before that is, how are cells finding their way to the dorsal midline? So we can think of different rules, different strategies of how cells know where it is and how they migrate to the dorsal midline. We have the first one here that we tried simulating, where cells, they know what the closest point of the dorsal midline is. And at the moment of convergence, they point their migration towards that closest point and move there in a straight line. So those simulations look a bit like this. The cells are spreading for the first 8 or 9 hours. And then at this moment, they will reconverge. And this gives a rather strange distribution here, where you get lots of cells on the poles of the dorsal midline, at the animal and vegetal poles here. And that's, of course, because if cells are moving towards the dorsal midline, which is here, all of the cells on the other side, their closest point is actually the pole. So all of the cells on this other part of the embryo are moving to the pole here, and the same on the other side. So another contrasting strategy could be that cells somehow they know the direction of east to west, we could say, on the embryo. So they're moving along these lines here. So if they do that, we can watch a simulation here of that again, where cells are distributing, and then at the point of convergence, they just are moving in a straight line. Well, it's in fact not a straight line, but they're moving in the east to west direction. That gives a distribution that looks like this, which you can see, of course, is very different to that original direction. So we learned that you might be, by looking at that distribution, you might be able to learn something about what rules the cells are using to find their way back to the dorsal midline. But the problem is that we don't know the full biology of the problem yet. So there is some evidence that chemotaxis is important. So there may be gradients forming throughout the embryo, but we don't know all of the details of that yet. And at this point, we kind of reached the limit of what we could find out with the modeling. And one reason for that was it was even difficult to measure this distribution here, because we were using traditional confocal microscopy, where we were obtaining these small sections of the embryos where we're in effectively 2 dimensions, where it was really a 3-dimensional problem. So what we've done now is we've turned to light sheet imaging here. We're teaming up with the lab, the Royer lab at the CZ Biohub, and we've obtained videos like these, where we can see the endoderm cells migrating during this point of development. And we're really at the point now where we're trying to segment these images and track the cells in their migration, so that we can make these more direct comparisons between the simulation results and experiment. So in conclusion, then, we can say we're quite convinced the simulations show that contact inhibition and combined with a long persistence length is a good strategy for dispersing cells and covering an embryo. And it has highlighted, the modeling's highlighted how these different strategies for finding the dorsal midline will give very different distributions of cells at the dorsal midline after reconvergence. So that's something we really want to explore in the data there. So our main to-do now really is to dig out these migration tracks from the light sheet data, so that we can make some more direct comparisons with that simulation. So that's everything. Thanks very much for listening, and to everyone who worked on this, and we're open for questions.
Thanks so much, Tom. Jocelyn, do you want to pick up from there? We have a couple of questions virtually, but I'm sure you've got some in the audience as well.
Sure. So we're first going to start with the expert modelers for the first 10 minutes, and then we're going to get into the questions from the audience, virtual and in the space. Stephanie, oh, would you come up, please? Yes. So now these are questions for Stephanie and Tom. You want to change the view? You want to change? Oh.
Jocelyn, just to let you know that we can't hear anything from the speakers.
Yeah, it is on.
Hello. Ooh,
Yes, we can hear you.
that's much better, isn't it?
Thanks, Brenda.
Okay, so I had a question about some of what's going on with the biology and decisions that were then made with modeling. So I can see in the modeling for, and you laid out beautifully how you sort of successfully added complexity to what the cells could do. And you're not explicitly invoking signaling in any way, but you're trying to capture it in these stochastic behaviors directionally, et cetera. Going back to what you told us about the biology, you mentioned this RAC1 signaling was responsible for the randomness and the migration. I'm wondering if you can tell us a little bit more about that. Is the RAC1 field uniform in the cell? Is RAC1 relatively scarce and it's local depletion that's leading to randomness? What is the nature of that randomness?
Yeah, so that's a great question. So in terms of what we know about the RAC1 in the cells, so just, for technical issues, we couldn't do a careful subcellular distribution of RAC1 activity. We do know that at this time point, the cells have relatively high RAC activity globally. And so given the known functions of RAC1 to promote actin polymerization, for example, so we think it's this globally high RAC1 that's driving this randomness, because it makes it more likely that you'll develop a leading edge on any edge of the cell. So you're just sort of increasing the probability that any part of the cell could become the leading edge. Does that answer your question?
Yeah, it does. So in that case, when you develop a leading edge, and I may have just missed it, does that mean the cell starts moving in that direction? And once it's selected, it's just going to keep moving that direction. It's not going to fluctuate.
Right. So yeah. So this is what. So right. So a cell develops a leading edge, and it pushes off in that direction. So what we think, this is speculation, but if you have globally high RAC activity everywhere in the cell, what that means is that the probability of that same leading edge persisting is relatively low. So you have a higher chance of another part of the cell just stochastically, developing a new leading edge, and now the cell is going to go off in that direction.
Okay. So the time scale on which the actin polarization, depolarization happens is sufficiently quick that you get this fluctuating definition of a leading edge.
Right. Yeah.
Okay.
Hi, can everybody hear me? Okay. Thank you, Stephanie and Tom, for a beautiful talk, beautiful data and modeling. So my question is more on the modeling side. I think one of the nice things about modeling is that you can sort of separate the process into these two parts, like what makes for best uniform dispersal of the cells, And I think, Tom, your work, sort of, you've nailed that a better. And then the second part is like, what are the ingredients for reaggregating? So for me, it was a little bit clear, and maybe you don't know from the biology, but like what the goal is for reaggregation. Do you think it's best for the cells to be uniformly spaced or aligned in this vertical strip in the end? And even if that were the answer, I know you showed different hypotheses for how they get there and how that affects the distribution. But in principle, you could separate that second part from the first part and just think about, placing the cells to different degrees of uniformity on the sphere, and then check how their initial conditions affect that. So I was wondering if you've thought systematically about that second part, and if you know what the goal is.
So from the modeling point of view for that second part, the very first and simplest thing we can do, and that we hope to get from the light sheet data, is just the distribution of cells along that dorsal midline. So after that reconvergence, how many cells have ended up where. And just that, we think from modeling could possibly fill the gap then, because we showed these two very different distributions, and we can say which one does it end up looking like, and it could give us a hint of what strategy the cells are using. But of course, potentially from the light sheet imaging, we can get the full tracks, so we can really see like in that first case where cells were trying to find, that they knew just the closest point in the dorsal midline and they just went straight there. We would see that directly if we can really extract those tracks from the data.
Thank you.
Sorry, I just saw a hand shoot up. Can you talk a little bit more about the rationale for choosing a fixed persistence length and assigning the stochasticity purely to the direction of travel?
Yeah, so we've chosen a fixed persistence length as just the simplest thing, because we don't know how it changes, and in fact we don't have a measure for what that persistence length is. So that's why we're exploring lots of the whole parameter space, really. I think it's something that's hard to get from the data, because you could see from Stephanie's data that the cells are so dense that they're colliding quite often. So actually really extracting a value for the persistence length, I think is quite a difficult challenge. And there's no obvious moment where persistence length changes. It doesn't strike you that it changes by eye, except for the moment when they all reconverge, because at that moment they move quite almost collectively in quite straight lines. Yeah, so for now we've done the simplest thing, which is to keep one persistence length.
And it makes a lot of sense. So the persistence length ends up being sort of a hidden variable. You did give us a typical travel speed. Do you have a sense of the variability of travel speed then from measurements?
No, that again, because they collide so often, it's also quite hard to get a variable speed. You can look at cells which are kind of right at the edge of the migrating cells, the sort of leaders, as it were, and they collide less frequently, and so from those, I guess it's easier to get a velocity, but otherwise you're extracting a velocity from quite a short motion, a short path. So again, we haven't tried, maybe Stephanie would know better, but we haven't tried to get an idea of the variation around that number, I think.
Yeah, I mean, we do have, data on both sort of instantaneous velocity as well as net displacement. But as Tom said, right, those numbers are include, you know, times that cells spend in collision with each other. So, so I should say, you know, I've been saying like, oh, there are these intrinsic parameters and these extrinsic parameters, but that's, you know, I'm fudging a little bit because even those intrinsic parameters, they, you know, we cannot, it's really hard to, like, as Tom said, find situations of a cell traveling without quickly bumping into another cell. There is one group that has done that. So they've been able to transplant endodermal cells and looked at migration in isolation. And they suggest that they still have this very low directional persistence. But these types of experiments are really very technically difficult and that have low throughput. And it's also, you know, you can argue, you know, when you do that, you're changing a lot about the cell and its environment. And so how physiologically relevant is that?
Do you have some pressing things? We want to leave some time for everyone, but
Real quick, wide-eyed, expand the scope question. So I think the elephant in the room is that this seems like a really stupid strategy for cells to get from a horizontal line to a vertical line.
Yes.
But because you have a nice model, you can actually test that, I would think, right? Maybe it's not stupid, right? So have you thought maybe from an evolutionary perspective, like whether there are completely different ways that cells can move that would achieve the goal more simply? Or maybe this is the simplest way to do it.
See what, Tom, can I take that? Yeah, so right, so I think what's happening is, right, the embryo, so initially the endodermal cells are specified in this, we can think of it as sort of a horizontal stripe, right, at the embryo margin, but then eventually the cells are going to span the anterior posterior length of the embryo. So you're turning, so in very simple terms, it's like you're turning a horizontal distribution to a vertical distribution. And so we think this dispersal is a way to, and we're hoping the model will bear us out, is a way to very quickly do that, right? And to do that without any gaps or discontinuities. And so I think zebrafish development relative to other organisms is very fast. So all of this happens in the matter of just a few hours. And so, you know, so what I would love to do is, you know, some sort of comparative thing and either look at, you know, related species that have slower development, and maybe in that case, the dispersal is not as important, or even take zebrafish embryos and maybe, you know, raise them at different temperatures and slow them down and see if that matters. So that's, but that is, I mean, that is completely, I have no basis for saying any of this. It's very hand wavy.
so we're definitely going to move on to the audience. Doesn't mean you guys can't chime in anymore. I'm going to ask a question, then Eric, and then we're going to go to Toby for some virtual questions. So I actually have two questions that are related. It seems to me you're modeling half of the entire phenomenon. You're modeling how the external ligand presented by the contacted cell is activating a receptor, turning off RAC1 to stop actin polymerization at that polarity site. But you're not modeling the reestablishment of polarity elsewhere. So I'm wondering if you're thinking about that. But the thing that I really am concerned about is you said that it is random in response to a question. I think Belinda's question, polarity is established randomly by globally active RAC1. But Tom definitely said that he is modeling it not that way. He's modeling it as opposite. It's definitely opposite.
Right. So in answer to your first question, so modeling, so right. So in terms of the leading edge polarization, it's known that there is this mutual antagonism between Rho A and RAC1. And so by definition, if you activate RAC1 in this side, it's going to, through this mutual antagonism, it's going to set up an opposite pole that, right. So they're sort of the same thing. So if you're saying I'm creating a back
Yeah.
here, that means that you're creating a front over here. So there.
So, but why not model it like you're seeing is if you say it's random, the new polarity side.
Oh, so, okay, so that, so, right, so it should be actually, and we, so this is in the date, so this is on Jesselyn's poster, so this is an advertisement for Jesselyn's poster. We actually do, if we look at the distribution of collision angles, we actually see that it tends to cluster around 180 degrees. So I think, you know, by these collision events activating, let's say, rho at the point of contact, you're probably setting up a leading edge on the opposite end.
Yeah, that I just to add, like we have explored that a tiny bit in the model where you can add when cells collide that they repolarize not exactly opposite each other, but there's some variation in that. So we've explored that at least a little, and at the moment it doesn't have any huge effect on our conclusions, but like, yeah, it's an important part of it.
Okay, so I'll follow up that with a somewhat related question, I think. So in the data from the mutants that Stephanie presented, it looked like they didn't aggregate, like they didn't form clumps, even though they were kind of doing this random motion. They were not, but in the simulations, right, the reason that you get such low heterogeneity is because of clumping, right? Because basically in that model, when cells sort of collide in a certain geometry, they can't get out because the model doesn't allow them to get out. But that may be a bit unphysical. So I'm just curious, or like unbiological. So I'm just curious if you've thought at all about, one, do you get aggregates in the data when you have random motion, like when they don't contact inhibit? Do you see those like islands of cells forming? And if you don't, how can you modify the model so that it sort of more accurately represents what happens when there isn't contact inhibition, maybe, if is that a thing?
So yeah, this is a great question. And Tom and I have, you know, we just had a conversation about this 2 days ago. So from the biology side, so you're right, they don't form these tight clumps or clusters. It's more like you have areas of like more dense, you know, the more density, and then areas of like low density. So irregularity in that way. And so that's because, so it's not, in addition to, you know, whether these interactions are inhibitory, you also have to think about whether these cells are physically capable of adhering to each other. And so we know we have expression data showing that they have not, at these stages, they have not turned on E-cadherin expression. So I think they just physically are incapable of like sticking to each other. And so maybe Tom can address this from the modeling side.
Yeah, I guess, yeah, just to add that there's also just a kind of perhaps an animation issue, or it might be that in the model that really you cells well, in the data, cells perhaps are stopping earlier than when they really touch each other, because their protrusions, even if they're not repolarizing, their protrusions are detecting other cells and stopping them moving. So they are making kind of aggregates perhaps, but they're not these tight balls of aggregates that you have in the modeling. And again, from the model point of view, that's a simple thing to solve, that you just need the distance at which you define a collision, basically, taking into account those protrusions.
Okay, thank you. Toby, I think we're ready for some questions on your end.
We've got a couple on Chi-Storm. I want to turn to Robert first. You had a nice one, Robert.
Well, thank both of you guys for a very nice talk. I'm curious what your model is based upon being a sphere, the number of sphere. So have you looked at all, see whether the behaviors are affected, whether you actually squish or you put pressure on top to make it, to deform the sphere to a bit more ellipsoid, and how that affects the behaviors you're observing?
So no, we haven't done that at all, but I'd love to do that. I'd love to do that both experimentally and in the model. I think it'd be really interesting. You completely change the problem. I guess that's what you're getting at, that you change that geometry, and so that could have a really interesting effect.
Thank you.
Really nice. Thanks, Tom, and thanks for the question, Robert. Well, one other perhaps, Matthew, you come on with a nice one, or perhaps you're.
He's in the audience.
He's in the audience. In that case, I may turn to Wallace for a final virtual one. Wallace, you had a grown culture question. No, I think he may have disappeared for an early lunch, in which case it is going to be Matthew in the audience.
Thanks. That was a great talk. So you mentioned that when trying to measure the velocities of cells, it's hard because they keep running into neighbors. I'm wondering if you can mask out cells based on how many neighbors they have. So try to just measure the velocities of the isolated cells or the ones that have.
Yeah, that would be a question for our collaborators who are doing the tracking for us.
Yeah, that would be a really useful thing to be able to do. Yeah. I think in the end, there's probably not lots of cells which are alone, you know, and definitely in the wild type case, because you have this quite dense and homogeneous distribution, there's not lots of cells that you can find isolated, but it'd still be super useful.
Hey, yeah, I really like the model, and I was thinking about from the dispersal to the midline. Have you guys tried integrating like some type of repulsion between cells? Like maybe they're a little less repulsive to each other, but still more attracted to the midline, so that when they get to the midline, they start to organize a little bit based on that, and they start to even out.
Yeah, that's a nice idea. So you mean if they arrive at the midline and they have a very inhomogeneous distribution, then that repulsion can sort of rectify that.
It could still be CIL, right?
Yeah, but
yeah,
Stephanie
yeah.
can, yeah.
Yes, yeah. So actually we think that CIL is not entirely turned off during the convergence event. So you can come ask me about that later.
Thank you.
Thank you.
Thank you. Thank you so much, Tom. We really